I just thought that was a kind of cool detail, right? But if there's lots of glucose around, this sequestration is inhibited by glucose. So it only does this when glucose is low and fructose 6-phosphate is high, which would be a situation where maybe glycogen is being broken down, right? Glucose is being used in other places. Okay? I thought that was a really slick way and a really unique way of controlling an enzyme. You're thinking about it? You don't think it's as cool as I think it is?
No, I just never heard of that. Like, so is that just the second way of preventing glycolysis from continuing? Because the first one was the glucose 6-phosphate if that builds up.
Right. But remember, we're talking different enzymes in different tissues. So this happens with hexokinase in all tissues. This happens with glucokinase in the liver. Okay.
But it's like one step further with after we
Right.
fructose 6-phosphate.
But what we'll see when we get there, glucose 6-phosphate and fructose 6-phosphate are in direct equilibrium with each other, so it really doesn't matter which one we're using for that control, because each one is affected by the other. Okay? Is that pretty slick? Okay. So, here's the reaction we've been talking about, right? Talking about hexokinase. So here is our glucose, right? Position 6, this hydroxyl out here off the ring. This is acting as the nucleophile. ATP is the electrophile. Hexokinase is the catalyst. These enzymes are often magnesium dependent, right? These kinase reactions often require magnesium as a cofactor, magnesium ions in the solution. Okay? Now, I can show you why we need magnesium, I think, right here. Now, they often show it this way, but if we go back to one of our organic chemistry nucleophilic substitution reactions. So say we wanted to do a substitution reaction with an alcohol leaving group. Right? Say we wanted to do a substitution reaction with an alcohol leaving group. And say we wanted to use a chloride ion as the nucleophile. Right? Straightforward SN2 type reaction. What's missing here? Evan, if you wear a purple shirt, I have to call it. What's missing here in order to get a successful nucleophilic substitution reaction?
You need a good leaving group.
We need a good leaving group. Do we have a good leaving group?
Not a standard.
No. A hydroxide ion as a leaving group stinks. It's too basic. How did we make it a better leaving group?
Protonate that side.
We protonate it, right? We have a hydrogen ion come in, and we protonate it. So, that hydrogen ion, if we think about it most globally, what is it? Well, we're going to make water as the leaving group, but what is the hydrogen ion acting as here?
Stabilizer?
Well, it's acting to
Stabilize
it, but let's go back to our fundamental organic chemistry definition. What's it acting as here? Well, hydrogen ions come from acids. Yeah, these are the hard questions, right? Hydrogen ions come from acids. So it's what kind of acid is it acting as?
Strongest.
What kind? What'd you say?
Lewis, no.
Lewis. Yes.
Oh.
Yeah. Right? It's acting as a Lewis acid.
Yeah, no.
What's the other thing that often acts as a Lewis acid? Metal ions. How did you get that? Right? So if we look here, in a lot of these reactions, the magnesium ion, there's a magnesium ion or some other divalent cation hanging around in these kinase reactions. Now, if you look at the talk in the enzyme chemistry, they talk about, the magnesium ions interacting and orienting the phosphates and all kinds of things. But remember, phosphoric acid is not one of our strong acids. It's a moderate acid. And when you deprotonate it, it gets to be, the next one is a weaker acid, a stronger base. So these phosphate leaving groups, while we talked about the resonance stabilization and all that stuff, they still have limitations as leaving groups. So if we're relating this back to our organic chemistry, one of the ways we can think about these divalent cations is these divalent cations are acting the same way the hydrogen ion is. That is, as a Lewis acid interacting with our basic leaving group to reduce some of the charge of the leaving group and thereby make it a better leaving group. So we can still relate this to what we learned from the basic mechanism of SN2 reactions that we learned in organic chemistry. Right? The same principles apply. Jen's thinking about that.
I was just wondering, in the book it said that the magnesium, they're not sure if it's like interacting with alpha, beta, or beta, wouldn't you want it, well, I guess you want the phosphate that's going to be attacked to be more stabilized. But I guess the leaving group, then you have one that's negatively charged, isn't that going to make it a bad leaving group?
Right, but this is where the debate comes in. Structurally, it looks like it may be in the initial enzyme structures, closer to this. But if you look, sometimes I think, when they showed the leaving group, they didn't show it here. And some of the ones I've looked at, they show the product as being ADP with a magnesium ion. So that's showing the magnesium ion interacting with the ADP leaving group. Like it could shift over. And that's where I think the debate comes. And in looking for this mechanistic information, I didn't see really good, clear descriptions of what the complete active site interactions were. So I think there is still some debate about, where does this electron density lie? Where is it interacting in the reactive complex? How's it interacting in the product complex? I think there's some question. But we can still go back and say, oh, at least one of the things it could be doing is helping our leaving group leave. Just like the hydrogen ion here helps our leaving group leave. Okay? Does that make sense?
Yeah.
Okay. Does that make sense? Kyle's thinking about it? Okay. Brooke, happy with that? We're good? Okay. I just downloaded this. This is showing some of the interactions. It doesn't show the interaction I was looking for. So I'm not going to use this. So this is the glucose, right? So this is the oxygen. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. This is just showing some of the hydrogen bonding interactions. What I was really looking for, right, is ultimately, and we'll do this in the next slide, in order to open this ring, this is something else. I'm dreaming. Ignore me. So, hexokinase. Hexokinase and glucokinase. This is the kinetics. Actually, I like this picture better. It's the same picture, but with reminding us where our normal fasting blood glucose is. This is taken from the IU School of Medicine. The Bloomington campus developed an online biochemistry tutorial, and it's really, really good. There's a link to it on ACE if you want to go look at it as another source for discussion of some of this stuff. And this was taken from that site. The medicalbiochemistrypage.org, right? That's why it says that across there. So we can see hexokinase, Km around 1 millimolar, right? Very steep. Normally we're well above that. This isn't showing the sigmoidal nature of glucokinase as much. It's very slightly sigmoidal. It just doesn't quite fit a Michaelis-Menten model. So this is sort of extended by that sigmoidal nature. Normal blood glucose is in the 5 to 7 millimolar range, so we're down here. So again, as blood glucose concentrations change from normal fasting concentrations to well-fed concentrations, what happens in most of the body? Yeah, not much, right? The rest of the body is, right, your red blood cells are pretty much transporting glucose at the same rate. Whether you just got out of bed and you're hungry after not eating all night, or whether you just ate a big pile of my mother's fluffy cloud mashed potatoes, right? Doesn't matter. This isn't changing rate much. What's the only tissue where you are going to get, well, the hexokinase itself isn't going to change. In muscle, you may get an increase in rate of transport of glucose into the muscle cell by upregulating the GLUT4 transporter. But once that glucose is in there, the rate of phosphorylation just doesn't change very much. Whereas in the liver, if you start at a very hungry 5 millimolar, you're at about what? And we're assuming this is, what is that, about a quarter, maybe 20% of Vmax. And by the time you get well-fed, overstuffed, have to unbutton the top button of your pants full, right? You're still at only about 80%, right? So Thanksgiving afternoon even, you still haven't saturated your glucokinase. Why is that important? And remember, where is glucokinase?
The liver.
The liver. And what's the liver's job? To regulate blood glucose concentrations. And the liver does not use glucose as a primary energy source. Right? The liver prefers not to use glucose as an energy source. It prefers to save it for other things. So, under normal conditions, actually probably the liver's hexokinase is working faster than its glucokinase. But as concentrations increase, the liver has the capacity to change its glucose utilization, where the rest of the tissues don't. Right? So the liver's rate of phosphorylation of glucose can increase so that the liver can directly participate in removing that extra glucose from the bloodstream to get it back to a normal level, below where actual toxicity can occur. Does that make sense? Are we happy with that? Okay? So that's the difference between glucokinase and hexokinase.
So if it got all the way full, it would be toxic.
Well, that's what happens in long-term diabetes. The problem is, remember, because glucose is a hemiacetal, that hemiacetal is always in equilibrium with an aldehyde. So there's some free aldehyde form of glucose at any instant. Aldehydes are very reactive toward nucleophiles, and amino groups are nucleophiles. So if glucose concentrations raise too high, you just get random reaction of glucose with amino groups, which can damage proteins, right? It changes the nature of protein interactions, damages proteins, and that's at least some of the source of damage when someone has uncontrolled diabetes. Part of the clouding of the lens of the eye is glucose reacting with proteins in the lens of the eye. By reacting with them, changing their three-dimensional structure, which then keeps them from transmitting light in the same way, and so the lens gets clouded. That's part of the reaction. you can, if you look at uncontrolled diabetics, if you take a sample of the protein from their blood serum, you can actually